The next topic is vector for gene transfer. First one, type plasmid. Type plasmid is the tumor inducing plasmid. Ty means tumor inducing. It produces crown gall disease in dicot plants. This is the structure of crown gall disease in dicot plants. Ty plasmid is present in soil bacterium, agrobacterium, tumefaciens. It is a circular double-stranded DNA and it contains 15,000 to 23,000 base pairs and it consists of different types of genes. First one, transfer gene for conjugation and opine catabolism gene for the degradation of opine and origin gene for replication and virulence gene for the transfer of tDNA and tDNA region. The type plasmid has an important DNA segment called tDNA. tDNA means transferred DNA. When an agrobacterium infects a plant cell, the tDNA becomes separated and integrated with the chromosome of the plant cell and cause crown gall tumor. Hence it is called tDNA. The tDNA consists of different types of gene. First one, border sequences at the two ends of tDNA, and the border sequences help the transfer of tDNA and open synthesize gene and cytokinin gene and auxin gene. The open gene synthesize open compounds which are used as energy source by the bacterium. And the cytokinin and auxin genes synthesize growth hormones which induces tumorous growth of the plant cell causing crown gall. When an agrobacterium infects a plant cell, the tDNA of the type plasmid becomes separated and integrated with the chromosome of the plant cell. The tDNA forms a part of plant genome and when the plant cell divides, the tDNA also divides. And the significance of type plasmid. The type plasmid is a cloning vector for plant cell. When a desired gene linked in the middle of tDNA, desired gene can be transferred to the plant genome when the bacterium infects the plant cell. When type plasmid is used as a cloning vector, there is no need for the species device to introduce the recombinant DNA into the host cell. And next one is riplasmid. Agrobacterium rhizogenes is a gram negative soil bacterium that incites hairy root disease in dicot plants. A plasmid called riplasmid is responsible for this disease. Riplasmid are analogous to agrobacterium tumefaciens type plasmid. Riplasmid organization. Large plasmid of 250 kilo base pair. Riplasmids are large plasmids of 250 kilo base pair and it has two tDNA fragments, transfer DNA and tDNA left and right separate by 15 kilo base pair segment and transfer DNA is similar to tDNA and it has gene for auxin and agropine synthesis. Agropine is the type of opine and tDNA has 4 genes, role A, role B, role C and role D. And role A is responsible for hairy root formation and role B is induces root initiation and callus formation, and role C promotes root growth, and role D suppresses callus growth. The rye plasmid as a virulence region that mediated tDNA transfer and an ori same as the Ti plasmid. This is the structure of rye plasmid. And uses of rye plasmid. Rye plasmid can be used as substitutes for Ti plasmid to transfer foreign genes to plants. And rye plasmid exchange DNA segments between plants and bacteria, and hence they could be steady gene exchange between the organisms, and they could be used to induce rooting in clones where rooting from callus or explant is difficult, and they could also be used to increase nodulation frequency of plantlets during micropropagation.